Close your eyes for a moment and imagine the vastness of space. Billions upon billions of stars scattered across an infinite darkness, each one a world unto itself. Now open them and consider this. Right now, at this very moment, you carry within you an ecosystem just as vast, just as complex, and just as wondrous as any galaxy. Trillions of microscopic organisms, more numerous than all the stars visible in the night sky, are living, breathing, and working inside your body. They coat your skin like stardust, line your digestive tract like constellations mapped across the heavens, and help orchestrate nearly every aspect of your health. This is your microbiome, the hidden universe inside you, and tonight we're going to explore it together. Welcome to Bedtime Biology, where science meets serenity. Each night, we explore the quiet wonders of the natural world, calming your mind and sparking your curiosity. Please, like the video, subscribe for more like this, and comment below with where you're checking in from. The hidden universe. Understanding what the microbiome is. Let's begin our journey by understanding what we mean when we talk about the microbiome. Picture yourself standing in a pristine forest. Everywhere you look, life teems in intricate harmony. Birds nest in the canopy above. Insects crawl along bark and beneath fallen leaves. Fungi network through the soil in invisible webs, and bacteria break down organic matter, returning nutrients to the earth. This forest is not just a collection of trees, it's an ecosystem, a community of organisms living together, each playing a role in the whole. Your body is remarkably similar. You are not just you, you are a walking ecosystem, a living habitat for an astonishing diversity of microscopic life. The term microbiome refers to all the microorganisms that live on and inside your body, along with all their genetic material. These residents include bacteria, which are by far the most numerous, but also fungi, viruses, archaea, and even tiny microscopic animals. Together, they form communities that are so abundant they actually outnumber your own human cells. Scientists estimate that the bacterial population alone ranges somewhere between 75 trillion and 200 trillion individual organisms. To put that in perspective, your entire body consists of about 50 trillion to 100 trillion human cells. You are, in a very real sense, more microbial than human. The microbiome is not a monolith. It varies dramatically depending on where you look. The community of microbes living in your mouth is entirely different from those residing in your gut, which are different again from those coating your skin. Each habitat within your body, each microenvironment, whether dry, moist, acidic, or oxygen-rich, fosters its own unique assembly of microbial species. Your gut alone is estimated to harbour between 500 and 1,000 different species of bacteria. The entirety of the human microbiome may consist of around 900 to 1,000 different species in total, though this number grows as scientists continue to discover new organisms that have never been cultured or studied before. The diversity is staggering. Research involving the Human Microbiome Project, a massive scientific effort that has sequenced samples from thousands of individuals, has revealed that the human gut contains more than 3,000 different bacterial species, spread across fewer than 130 genera. This means that the diversity of bacteria in your intestines is concentrated at the species and strain level. Even more remarkable, while many of these species are shared across people, the specific strains and their relative abundances can vary widely from person to person. Your microbiome is as unique as your fingerprint, shaped by your genetics, your environment, your diet, and your life history. What makes the microbiome even more fascinating is its genetic richness. The collective genes of all the microbes in your gut, known as the microbiome or metagenome, are estimated to contain around 3 million genes. That's about 150 times more genes than are found in the human genome. This staggering genetic diversity means that your microbes provide you with an enormous reservoir of metabolic capabilities, functions that your own cells simply cannot perform. They contribute enzymes and biochemical pathways that allow you to break down complex plant fibers, synthesize essential vitamins, produce neurotransmitters, and defend against harmful pathogens. In essence, your microbiome expands what it means to be human, extending your biological capabilities far beyond what your own DNA can code for. The relationship between you and your microbiome is ancient. For millions of years, mammals and their commensal microorganisms have co-evolved together, developing a mutualistic relationship where both parties benefit. The microbes gain a stable environment, protection from the outside world, and a steady supply of nutrients. In return, they perform essential services for their host. They help digest food, synthesize vitamins, train the immune system, and even protect against colonization by harmful bacteria. This partnership is so deeply ingrained that neither you nor your microbes could thrive without the other. You are not separate from these microorganisms. You are, in the truest sense, a superorganism, a blend of human and microbial cells working together as one. But the story doesn't end with bacteria. The microbiome also includes fungi, which may not be as abundant as bacteria, but are nonetheless important players. Fungi like Candida and Malassezia inhabit various parts of your body, particularly your skin and gastrointestinal tract. They contribute to the ecosystem's balance and can influence your health, sometimes becoming problematic when they overgrow. Viruses, too, are part of your microbiome. These include bacteriophages, viruses that infect bacteria, which are extraordinarily abundant and play a role in regulating bacterial populations. Some scientists have even discovered novel types of viroid-like elements, called obelisks, within the human microbiome, along with their associated proteins, called oblins, adding yet another layer of complexity to this microscopic world. Archaea, another group of microorganisms, are also present, though in much smaller numbers than bacteria. These ancient single-celled organisms are particularly adapted to extreme environments, but they also make their home in your gut, contributing to processes like methane production. And then there are the micro-eukaryotes, tiny protozoans, and even microscopic amoebas that live peacefully in your gastrointestinal tract, coexisting with the rest of the microbial community. When scientists first began studying the microbiome, they thought they might find a core microbiome, a set of bacterial species that everyone shares. Culture-based studies suggested this might be true. For example, Escherichia coli, a bacterium familiar to most people, can be isolated from nearly everyone. But as DNA sequencing technologies advanced and researchers began to study the microbiome without needing to culture bacteria in the lab, a different picture emerged. Instead of a universal core, they found vast diversity. While certain bacterial groups are common across people, the specific species and strains vary tremendously. Two healthy people might share very few of the exact same bacterial species, yet both can have perfectly healthy microbiomes. This is because what matters most is not necessarily which species you have, but what those species are doing, their functional capabilities. This brings us to an important distinction: taxonomic diversity versus functional diversity. Taxonomic diversity refers to the variety of species present, the different names on the microbial census. Functional diversity, on the other hand, refers to the jobs those species perform. Research has shown that while the taxonomic makeup of the microbiome can differ substantially between individuals, the functional capacity tends to be more conserved. In other words, different species can perform similar roles. Imagine a warehouse staffed by different workers. The specific people might change, but as long as each job gets done, packaging, shipping, inventory, the warehouse runs smoothly. The microbiome works much the same way. Different species can metabolize fiber, produce vitamins, or defend against pathogens. And as long as these functions are covered, the microbiome can remain healthy and balanced. This functional redundancy is one reason why the microbiome is so resilient. It can adapt to changes, compensate for the loss of certain species, and maintain stability even in the face of perturbations. But this resilience has limits. When the balance is disrupted too severely, whether by antibiotics, poor diet, chronic stress, or illness, the microbiome can shift into a state called dysbiosis, where harmful or opportunistic species proliferate and beneficial functions are lost. We'll explore dysbiosis in more detail later, but for now, it's enough to understand that the health of your microbiome depends on maintaining a rich diversity of species and a robust set of functional capabilities. The study of the microbiome is still relatively young. It wasn't until the mid-1880s that scientists first confirmed that microorganisms are a normal part of the human system. An Austrian pediatrician named Theodor Escherich observed bacteria in the intestinal flora of both healthy children and those affected by diarrheal disease. That bacterium was later named Escherichia coli in his honor. In the decades that followed, scientists isolated and characterized more microorganisms from the human body, including Veillonella parvula from the oral and digestive tracts in 1898, and bifidobacteria from the intestines in 1900. Throughout the 20th century, researchers continued to identify and study microbes from the skin, nasal passages, oral cavities, gastrointestinal tract, and urogenital tract, slowly building a picture of the microbial communities that call us home. But it was the advent of high-throughput DNA sequencing in the early 21st century that truly revolutionized our understanding. No longer did scientists need to culture bacteria in the lab, a process that often failed because many bacteria are notoriously difficult to grow outside their natural environment. Instead, they could extract DNA directly from samples and sequence the genes present, particularly the 16S ribosomal RNA gene, a genetic marker found in all bacteria that allows scientists to identify and classify them. This technology opened the floodgates. Suddenly, researchers could see the full extent of microbial diversity, discovering thousands of species that had never been cultured before. Projects like the Human Microbiome Project and MetaHit began mapping the microbial communities of healthy individuals, creating reference databases and revealing patterns of diversity and function across different body sites and populations. These large-scale efforts have shown us that the microbiome is not static. It changes over time, shifting with age, diet, geography, lifestyle, and health status. The microbiome of an infant is very different from that of an adult, and the microbiome of a person living a traditional hunter-gatherer lifestyle is different from that of someone living in a modern industrialized city. These differences have profound implications for health. Populations with higher microbial diversity tend to be healthier, while reduced diversity has been linked to a range of conditions, including obesity, inflammatory bowel disease, diabetes, allergies, and even mental health disorders. As we drift deeper into this exploration, keep in mind the scale of what we're discussing. Every moment, as you breathe, as you digest your last meal, as you rest and dream, trillions of microscopic organisms are at work inside you. They are breaking down food, synthesizing molecules, communicating with your immune system, and even influencing your brain. They are ancient partners, evolved alongside us, embedded in our biology so thoroughly that we cannot separate their story from our own. To understand the microbiome is to understand a hidden dimension of yourself, an invisible universe that shapes who you are in ways we are only beginning to comprehend. Now that we've established what the microbiome is, let's take a journey through your body and explore the diverse microbial communities that inhabit its many landscapes. A journey through your body. Microbiomes of different body sites. Your body is not one uniform environment. It is a patchwork of distinct ecosystems, each with its own climate, terrain, and resources. Just as you would find different plants and animals in a desert than in a rainforest, you find different microbial communities in different parts of your body. Each site, each micro-environment selects for microbes best adapted to thrive there. Tonight, we'll travel through these habitats, starting with your skin, moving inward to your mouth, descending into your gut, and even touching on other less explored regions. As we go, picture these places not as sterile surfaces, but as thriving ecosystems, alive with microscopic activity. Let's begin with your skin, the body's largest organ, spanning roughly 2 square meters in an average adult. Your skin acts as the primary barrier between your internal world and the external environment. It's a harsh landscape for microbes. The surface is dry, acidic, salty, and constantly exposed to the elements. Ultraviolet light from the sun, fluctuations in temperature and humidity, the friction of clothing, the chemicals in soaps and lotions, all of these factors make the skin a challenging place to live. And yet, it is colonized by an astonishing diversity of microorganisms. Bacteria, fungi, viruses, and even tiny mites inhabit your skin, forming communities that vary from one body site to another. The skin can be divided into different microenvironments based on its physical properties. Some areas are sebaceous or oily, particularly the face, chest, and back, where sebaceous glands produce sebum, a lipid-rich secretion. These oily regions tend to be dominated by lipophilic bacteria, organisms that thrive in the presence of fats, such as species of Propionibacterium and Corynebacterium. Other areas are moist, like the armpits, the groin, the spaces between your toes, and the folds of your elbows and knees. These humid zones harbor bacteria that prefer moisture, including species of Staphylococcus and Corynebacterium. Then there are the dry areas, such as the forearms, the palms of your hands, and much of the rest of your body's surface. These drier regions support a more diverse array of bacteria, with communities that can shift more dramatically over time. In total, scientists have identified around 1,000 different species of bacteria that can inhabit human skin, spread across 19 different phyla. Most of these bacteria fall into 4 major groups: actinobacteria, bacteroidetes, firmicutes, and proteobacteria. But within those broad categories, the specific species and strains can vary widely. Studies have shown that even a small area, like the palm of your hand, can harbour more than 150 different bacterial species. Remarkably, only about 17% of those species are shared between your 2 hands, and only about 13% are shared between different people. This means that your skin microbiome is highly individual, shaped by your genetics, your environment, your hygiene habits, and countless other factors. The skin microbiome is not just passively sitting on your surface. It's actively interacting with your skin cells and immune system. Some skin bacteria produce antimicrobial compounds that help ward off pathogenic invaders. For example, certain strains of Staphylococcus epidermidis, a common skin commensal, secrete molecules that inhibit the growth of Staphylococcus aureus, a pathogen that can cause infections. The skin microbiome also helps maintain the slightly acidic pH of your skin, which many harmful bacteria find inhospitable. And it plays a role in training your immune system, teaching immune cells to distinguish between harmless commensals and dangerous pathogens. Fungi are another important component of the skin microbiome. The most common fungal residents belong to the genus Malassezia, which can account for up to 80% of the fungi on your skin, depending on the body site. Malassezia species consume oils secreted by your sebaceous glands, and while they are usually harmless, they can sometimes contribute to conditions like dandruff and seborrheic dermatitis. Other fungi, as well as viruses and even tiny parasitic mites, called demodex, also inhabit the skin. These mites live in hair follicles and sebaceous glands, feeding on skin oils and dead cells. While they have been implicated in skin conditions like rosacea, they are also found on healthy skin, suggesting they may be part of the normal commensal community. One of the most interesting findings about the skin microbiome is how variable it can be over time. Compared to the relatively stable microbiome of the gut, the skin microbiome shifts more frequently, influenced by factors like washing, sweating, exposure to different environments, and even the clothes you wear. Sites that are more occluded, like the inside of your ear or your nostrils, tend to be more stable, while exposed areas like your hands and forearms can change significantly from day to day. Despite this variability, your overall skin microbiome tends to remain relatively consistent in its broad composition, a phenomenon known as individuality. You carry a core set of microbial signatures that are uniquely yours, even as specific species come and go. Now, let's move inward to your mouth. The oral cavity is one of the most diverse microbial habitats in the body. Every surface, your teeth, your tongue, your gums, the inside of your cheeks, the roof of your mouth, harbours its own distinct microbial community. In total, your mouth is home to several hundred different bacterial species, and recent estimates suggest that there may be even more diversity than previously thought. The Human Microbiome Project found that oral sites, along with the gut, showed the greatest microbial richness compared to other body sites. The mouth presents a unique set of conditions for bacteria. It's a warm, moist environment with a steady supply of nutrients from the food and drink you consume. Saliva bathes the mouth constantly, providing water, minerals, and proteins, while also washing away debris and bacteria. The pH of your mouth fluctuates depending on what you eat, becoming more acidic after you consume sugars or carbohydrates, then gradually returning to a more neutral state. Oxygen levels also vary, higher at the surface of your tongue and cheeks, lower in the crevices between your teeth, and below the gum line. These varying conditions create numerous microbial niches. The bacteria that thrive on the smooth surfaces of your teeth, forming the sticky biofilm known as plaque, are different from those that colonize the rough surface of your tongue, or the pockets around your gums. Streptococcus mutans, for example, is a common inhabitant of dental plaque. It metabolizes sugars and produces acid as a byproduct, which can erode tooth enamel and lead to cavities. Other bacteria, like species of Veillonella, help neutralize acids, protecting your teeth. The balance between these different species can determine whether you develop cavities or maintain healthy teeth. The oral microbiome also plays a role in your overall health beyond your mouth. Some oral bacteria have been linked to systemic conditions. For instance, certain species have been found in atherosclerotic plaques, suggesting a potential connection between oral health and cardiovascular disease. The mechanisms are still being studied, but it's thought that oral bacteria may enter the bloodstream through small injuries in the gums, triggering inflammation elsewhere in the body. This is one reason why maintaining good oral hygiene is so important, not just for your teeth, but for your whole body. From your mouth, we travel down your esophagus and into your stomach. The stomach is an inhospitable place for most microbes. It's highly acidic, with a pH that can drop as low as 1 or 2, a level that would kill most bacteria. For many years, scientists believed the stomach was essentially sterile. That view changed in the 1980s with the discovery of Helicobacter pylori, a bacterium that has evolved to survive in the stomach's harsh environment. H. pylori produces an enzyme called urese that neutralizes stomach acid around it, creating a more favorable microenvironment. While H. pylori is present in about half the world's population, it can also cause problems, leading to ulcers and increasing the risk of stomach cancer in some people. This is a prime example of how a commensal organism can become a pathogen under certain conditions. Below the stomach, we reach the small intestine, where digestion and nutrient absorption primarily occur. The small intestine has a relatively lower microbial density compared to the colon, but it's still colonized by bacteria, particularly in the lower section, the ileum. The bacteria here help break down nutrients that escaped digestion higher up, produce vitamins, and interact with the gut-associated lymphoid tissue, a critical component of the immune system. And then we arrive at the colon, the large intestine, the microbial metropolis of your body. This is where the vast majority of your gut bacteria reside. The colon is a long, tube-like structure, and as you move from the beginning to the end, the microbial communities change. The environment in the colon is largely anaerobic, meaning there's very little oxygen, and the bacteria that thrive here are specially adapted to these low oxygen conditions. The colon is also home to an enormous amount of undigested material, dietary fibers, and complex carbohydrates that your own digestive enzymes cannot break down. This is where your microbes shine. The bacterial composition of the colon is dominated by a few major phyla, primarily Bacteroidetes and Firmicutes, though Proteobacteria, Actinobacteria, Verrucomicrobia, and Fusobacteria are also present. Within these phyla, there are hundreds of species and thousands of strains. Some of the most important gut bacteria belong to genera like Bacteroides, Prevotella, Faecalibacterium, Clostridium, Bifidobacterium, and Ruminococcus. These bacteria perform a staggering array of functions. They ferment dietary fibers, producing short-chain fatty acids like butyrate, acetate, and propionate, which are absorbed and used by your body. They synthesize vitamins, including vitamin K and several B vitamins. They break down complex plant polysaccharides, proteins, and even some fats. They produce metabolites that communicate with your immune system, helping to maintain tolerance and prevent inflammation. They even influence the production of neurotransmitters, chemicals that affect your brain. The gut microbiome is not evenly distributed. Different sections of the colon have different bacterial communities, and the bacteria that live in the mucus layer, close to the intestinal wall, are different from those suspended in the lumen, the central cavity of the gut. This spatial organization is important for maintaining a healthy barrier between your microbes and your immune system. The mucus layer acts as a buffer, preventing bacteria from directly contacting the epithelial cells that line your gut. Specialized immune cells patrol this border, sampling bacterial products and maintaining a delicate balance between tolerance and defense. Your gut microbiome is also influenced by what you eat. Different diets support different bacterial communities. A diet high in fiber, for example, promotes the growth of fiber-fermenting bacteria like Prevotella, which are abundant in populations that consume traditional plant-rich diets. In contrast, a Western diet high in fat and sugar and low in fiber tends to favor a different set of bacteria, including some that produce pro-inflammatory metabolites. These diet-induced shifts in the microbiome can have profound effects on your health, influencing everything from your weight to your risk of chronic diseases. Beyond the gut, other parts of your body also harbor microbial communities. The urogenital tract, for example, has its own microbiome. In women, the vaginal microbiome is typically dominated by lactobacillus species, which produce lactic acid that keeps the pH low and helps prevent infections. The composition of the vaginal microbiome can shift depending on factors like menstrual cycle, pregnancy, and hygiene practices. In men, the urogenital microbiome is less well studied, but also includes a variety of bacterial species. Even your respiratory tract, your nose, throat, and lungs, is colonized by microbes. The nasal passages harbor bacteria that can influence your susceptibility to respiratory infections. And recent research has shown that even the lungs, once thought to be sterile, contain a small but significant microbial community. These respiratory microbes may play a role in immune function and lung health. As we conclude this tour of your body's microbial habitats, take a moment to appreciate the sheer diversity and complexity of what you carry with you. Every surface, every cavity, every fold of your body is home to a unique ecosystem. These microbes are not invaders. They are residents, ancient partners that have shaped and been shaped by your biology. They protect you, nourish you, and communicate with your cells in ways we are only beginning to understand. In the next section, we'll explore how this incredible community first takes root, beginning with the moment you are born. The birth of your microbiome, from delivery to development. Imagine the moment of birth. After months of growth in the relative sterility of the womb, a newborn enters the world and is suddenly bathed in microbes. This is the beginning of the microbiome, the first seeding of the microbial communities that will accompany the infant throughout life. how this initial colonization occurs and the factors that shape it can have lasting effects on health and development. Tonight, we'll trace the journey of the microbiome from birth through infancy and into adulthood, exploring how it is established, how it matures, and how early disruptions can influence lifelong health. For many years, scientists believed that the womb was a sterile environment, that babies were born germ-free and only encountered microbes after birth. Recent research has challenged this view, with some studies suggesting that bacteria may be present in the placenta, amniotic fluid, and even the fetus itself. However, this remains a topic of debate, with other studies finding that these apparent bacteria may actually be contaminants or artifacts of sample collection. What is clear, though, is that the moment of birth represents a massive influx of microbial exposure, far greater than any that might occur in utero, and that this initial exposure profoundly shapes the infant's developing microbiome. The mode of delivery, vaginal birth versus caesarean section, is one of the most significant factors influencing the early microbiome. During vaginal delivery, the infant passes through the birth canal and is exposed to the mother's vaginal and fecal microbiota. The vagina is rich in lactobacillus species, bacteria that ferment sugars and produce lactic acid. And these bacteria, along with others from the maternal gut and perineal area, colonize the infant's skin, mouth, and gut. Studies have consistently shown that vaginally delivered infants harbor bacterial communities that resemble their mother's vaginal microbiota, particularly in the first few days and weeks of life. Caesarean section, on the other hand, bypasses the birth canal entirely. Infants born by caesarean are instead exposed first to the bacteria of the operating room and the skin of the healthcare workers and parents. As a result, their early microbiome is often dominated by skin-associated bacteria like Staphylococcus, Corynebacterium, and Propionibacterium, rather than the Lactobacillus and Bacteroides, typical of vaginally delivered infants. This difference in early colonization is not trivial. Multiple studies have found that caesarean-born infants have delayed establishment of certain key bacterial groups, particularly Bifidobacterium and Bacteroides, which are important for immune development and metabolic programming. The implications of these differences extend beyond the microbiome itself. Epidemiological studies have linked caesarean delivery with an increased risk of certain health conditions later in life, including asthma, allergies, obesity, type 1 diabetes, and inflammatory bowel disease. While correlation does not equal causation, and many factors contribute to these conditions, the altered microbiome is thought to be one potential mechanism. The immune system undergoes critical training in early life, learning to distinguish between harmless commensals and dangerous pathogens. If this training is disrupted because key bacterial species are missing or delayed, the immune system may develop improperly, leading to increased susceptibility to allergic and autoimmune diseases. This has led to considerable interest in strategies to restore the microbiome of caesarean-born infants. One such strategy is vaginal seeding, also called vaginal microbial transfer, where infants delivered by caesarean are swabbed with gauze soaked in the mother's vaginal fluids immediately after birth. The idea is to artificially introduce the vaginal microbiota that the infant would have encountered during vaginal delivery. Early studies, including a pilot trial published in 2016, showed that vaginal seeding could partially restore the microbiome of caesarean-born infants. bringing their bacterial communities closer to those of vaginally delivered babies, particularly in the mouth and on the skin. However, more recent and larger studies have yielded mixed results. Some have found that vaginal seeding does increase levels of certain bacteria, particularly lactobacillus, in the early weeks of life. Others have found little to no effect on the gut microbiome, especially beyond the first month. One study found that oral administration of maternal vaginal microbes did not significantly alter gut microbiome development in cesarean-born infants, and that the limited colonization by vaginal strains suggested that other sources, such as the maternal perianal area or skin, might play a larger role in seeding the neonatal gut. Another trial found that while vaginal seeding did influence the microbiome, the effect was modest and short-lived, with differences diminishing over time. The safety of vaginal seeding has also been questioned. There is a potential risk of transmitting infections, including Group B streptococcus, herpes simplex virus, or sexually transmitted infections from mother to infant. For this reason, the American College of Obstetricians and Gynecologists does not currently recommend vaginal seeding outside of research settings. Alternative approaches, such as probiotic supplementation, may offer safer and more reliable methods to support healthy microbiome development in cesarean-born infants. Regardless of delivery mode, the microbiome continues to develop rapidly in the first months and years of life. Breastfeeding is one of the most important factors shaping this development. Breast milk is not just a source of nutrition, it's also a complex mixture of prebiotics, components that promote the growth of beneficial bacteria, and probiotics, live bacteria themselves. Human milk contains oligosaccharides, complex sugars that the infant cannot digest but that serve as food for gut bacteria, particularly Bifidobacterium species. These bacteria are especially well adapted to metabolize milk oligosaccharides, and they thrive in the guts of breastfed infants. Studies have shown that the gut microbiome of breastfed infants is typically dominated by Bifidobacterium, along with other beneficial species, and tends to be more stable and less diverse than that of formula-fed infants. Formula-fed infants, in contrast, often have more diverse microbiomes earlier in life, with higher levels of bacteria from the Bacteroidetes and Proteobacteria phyla. While both breastfed and formula-fed infants can grow up healthy, breastfeeding is associated with certain health benefits, including lower rates of infections, allergies, and obesity, which may be partly mediated by the microbiome. As the infant grows and begins to consume solid foods, the microbiome undergoes a major transition. The introduction of complementary foods, typically around 6 months of age, brings a flood of new nutrients and challenges. The microbiome diversifies, incorporating new bacterial species capable of breaking down the complex carbohydrates, proteins, and fats found in solid foods. This dietary transition is gradual, and by the time a child is around 2 to 3 years old, their gut microbiome begins to resemble that of an adult in structure and function, though it continues to mature throughout childhood. This early period of life, particularly the first 1,000 days from conception to age 2, is considered a critical window for microbiome development. During this time, the microbiome is highly plastic, responsive to environmental influences, and it's establishing the patterns that will persist into adulthood. Disruptions during this window, such as antibiotic exposure, poor diet, or stress, can have lasting effects. Antibiotics, in particular, can dramatically reduce microbial diversity, and if given repeatedly or early in life, they may prevent the normal maturation of the microbiome. Studies in mice have shown that early life antibiotic exposure can lead to persistent changes in the microbiome and increased susceptibility to metabolic and immune disorders. The composition of the early microbiome is also influenced by factors beyond diet and antibiotics. Living environment plays a role. Infants growing up in homes with pets, especially dogs, tend to have more diverse microbiomes, likely due to increased exposure to different bacteria from the environment. Hygiene practices, though important for preventing infections, can also influence microbial exposure. The so-called hygiene hypothesis suggests that reduced exposure to microbes in overly sanitized environments may contribute to the rise of allergic and autoimmune diseases in developed countries, though this is still an area of active research. Geography and lifestyle have profound effects on the microbiome. Studies comparing the microbiomes of people from different parts of the world have revealed stark differences. Populations living traditional hunter-gatherer or agrarian lifestyles, such as the Hadza people of Tanzania, have microbiomes that are much more diverse than those of people living in industrialized Western societies. The Hadza, for example, consume between 100 and 150 grams of dietary fiber per day, more than 10 times the amount eaten by the average person in the United States. This high-fiber diet supports a rich community of fiber-fermenting bacteria, particularly species from the genus Prevotella, which can make up to 60% of the gut microbiome in non-Western populations, but are much less abundant in Western populations. These differences are not just academic curiosities, they have real implications for health. Higher microbial diversity is generally associated with better health outcomes, while reduced diversity has been linked to conditions like obesity, inflammatory bowel disease, type 2 diabetes, and autoimmune disorders. As societies industrialize, adopting Western diets and lifestyles, their microbiomes shift, often becoming less diverse. This loss of microbial diversity is sometimes referred to as the disappearing microbiota hypothesis, and it's thought to be one factor contributing to the rise of chronic diseases in modern populations. Animal studies have provided compelling evidence for how diet can shape the microbiome across generations. In one experiment, mice colonized with human gut bacteria were fed a low-fiber diet. Over the course of several generations, the mice progressively lost microbial diversity. When the offspring of these mice were returned to a high-fiber diet, some of the lost species could be restored, but after 4 generations on a low-fiber diet, the missing bacteria were gone for good, unable to be recovered even with dietary intervention. This suggests that the erosion of microbial diversity may not be easily reversible, and that maintaining a diverse microbiome requires sustained effort, particularly in terms of diet. As we think about the birth and development of the microbiome, it's important to recognize that this is not a deterministic process. While early life sets the stage, the microbiome remains dynamic throughout life, influenced by ongoing diet, lifestyle, health, and environmental exposures. The adult microbiome is relatively stable compared to the rapid changes of infancy, but it still shifts in response to major dietary changes, antibiotic use, illness, travel, and aging. In the elderly, the microbiome can become less diverse and less stable, which may contribute to age-related health decline. The story of the microbiome's development is one of remarkable plasticity and resilience, but also vulnerability. The early establishment of a healthy, diverse microbiome lays a foundation for lifelong health. Disruptions during critical windows can have lasting consequences, but the microbiome's adaptability means that it's never too late to support it through healthy habits. In the next section, we'll explore one of the most fascinating aspects of the microbiome, its connection to the brain. The gut-brain axis. How microbes shape your mind. The idea that your gut could influence your mind might sound strange at first. We tend to think of the brain as the seat of consciousness, emotion, and thought, separate from the digestive system. But scientists have discovered that the gut and brain are in constant communication, exchanging signals through a complex network of nerves, hormones, and biochemical messengers. This bidirectional highway is known as the gut-brain axis, and the gut microbiome plays a central role in how this communication works. Tonight, we'll explore this remarkable connection and discover how the microscopic residents of your gut can influence your mood, your behavior, and even your mental health. The gut-brain axis is not a new concept. As far back as the early 20th century, scientists like Pavlov recognized that emotional states could affect digestion, that nervousness could lead to an upset stomach. But the idea that the gut could also influence the brain was slower to develop. It wasn't until 2004 that a landmark study on germ-free mice, animals raised in completely sterile conditions without any microbes, revealed just how important the microbiome is for brain function. These germ-free mice showed an exaggerated stress response compared to normal mice with intact microbiomes. When exposed to stress, they released abnormally high levels of stress hormones, suggesting that the absence of gut bacteria had altered the development and function of their stress response system. Since then, a growing body of research has confirmed that the gut microbiome influences many aspects of brain function and mental health. The mechanisms are complex and multifaceted, involving several major pathways: the nervous system, the endocrine system, the immune system, and the production of microbial metabolites. Let's start with the nervous system. The gut is sometimes called the second brain because it contains an extensive network of neurons known as the enteric nervous system. This network can operate independently of the brain, controlling many aspects of digestion without input from the central nervous system. But the enteric nervous system is also connected to the brain via the vagus nerve, a major nerve that runs from the brainstem down through the neck and chest into the abdomen. The vagus nerve is a two-way communication line. Signals travel from the brain to the gut, influencing digestion and gut motility, and signals travel from the gut back to the brain, carrying information about the state of the digestive system. Importantly, the microbiome can influence these signals. Gut bacteria can produce or modulate neurotransmitters, the chemical messengers that neurons use to communicate. For example, certain bacteria produce gamma-aminobutyric acid, or GABA, an inhibitory neurotransmitter that helps calm neural activity. Others produce serotonin, a neurotransmitter critical for mood regulation. In fact, the vast majority of the body's serotonin, about 90 to 95%, is produced in the gut, not in the brain. While most of this gut-derived serotonin does not cross the blood-brain barrier and act directly on the brain, it influences gut function and sends signals to the brain via the vagus nerve. Some researchers believe that alterations in gut serotonin production driven by changes in the microbiome, could contribute to mood disorders like depression and anxiety. The endocrine system also plays a key role in the gut-brain axis. The hypothalamic pituitary adrenal axis, or HPA axis, is the body's main stress response system. When you experience stress, the hypothalamus in your brain releases a hormone that signals the pituitary gland, which in turn signals the adrenal glands to release cortisol, the primary stress hormone. The gut microbiome can influence the activity of the HPA axis. Studies in germ-free mice have shown that without a microbiome, the HPA axis becomes hypersensitive, leading to exaggerated stress responses. Colonizing these mice with normal gut bacteria can partially restore normal HPA function, demonstrating that the microbiome is involved in calibrating the stress response during early development. The immune system provides another major pathway for gut-brain communication. The gut is home to a large portion of the body's immune system, and the microbiome is constantly interacting with immune cells. These interactions produce signaling molecules called cytokines, which can influence brain function. Pro-inflammatory cytokines, such as interleukin-6 and tumor necrosis factor alpha, can cross the blood-brain barrier or send signals to the brain via other routes, where they affect mood, cognition, and behavior. Chronic low-grade inflammation, often driven by an imbalanced gut microbiome, has been linked to depression, anxiety, and other mental health disorders. This has led to the hypothesis that some cases of depression may be partly driven by gut-derived inflammation. Beyond neurotransmitters and immune signals, gut bacteria produce a vast array of metabolites, small molecules that can enter the bloodstream and travel throughout the body, including to the brain. Short-chain fatty acids, particularly butyrate, propionate, and acetate, are produced when gut bacteria ferment dietary fibers. These SCFAs have numerous beneficial effects. They provide energy to the cells lining the gut, help maintain the integrity of the gut barrier, and have anti-inflammatory properties. But they also influence the brain. SCFAs can modulate the blood-brain barrier, affect the production of neurotransmitters, and influence the activity of microglia, the immune cells of the brain. Animal studies have shown that SCFAs can affect behavior, reducing anxiety-like behavior and even improving memory and learning. Other microbial metabolites also play roles in brain function. Tryptophan, an amino acid obtained from the diet, can be metabolized by gut bacteria into various compounds, some of which can cross into the brain and influence neurotransmitter systems. Indole, a metabolite produced by certain gut bacteria, can activate the aryl hydrocarbon receptor, a protein involved in immune regulation and nervous system development. Researchers are still working to map out all the metabolites produced by the gut microbiome and understand how each one affects the brain. But it's clear that the chemical crosstalk between gut microbes and the nervous system is incredibly rich and complex. The connection between the gut microbiome and mental health is not just theoretical. Clinical and epidemiological studies have found associations between gut microbiome composition and various psychiatric disorders. In people with major depressive disorder, for example, some studies have reported reduced microbial diversity and alterations in specific bacterial genera. Certain genera, such as Faecalibacterium and Coprococcus, which are associated with the production of beneficial metabolites, have been found to be depleted in people with depression. Conversely, some studies have found increased levels of pro-inflammatory bacteria. Similar patterns have been observed in anxiety disorders, autism spectrum disorder, bipolar disorder, and schizophrenia, though the specific changes vary, and more research is needed to establish causality. One of the most compelling lines of evidence comes from studies of probiotics, often called psychobiotics, when they are used to target mental health. In animal studies, certain probiotic strains have been shown to reduce anxiety-like and depression-like behaviors. For example, Lactobacillus rhamnosus has been found to reduce stress-induced corticosterone and anxiety-like behavior in mice. In human studies, results have been more mixed, but still promising. Some trials have found that probiotic supplementation can improve mood, reduce anxiety, and even enhance cognitive function in healthy individuals and in people with depression. One study found that a probiotic containing bifidobacterium and lactobacillus strains improved symptoms of depression in patients to a similar degree as conventional antidepressant medications, though more rigorous and larger trials are needed to confirm these findings. The gut microbiome may also influence neurodevelopmental disorders. Autism spectrum disorder, for instance, has been associated with gastrointestinal problems in many patients, and several studies have found differences in the gut microbiome of individuals with autism compared to neurotypical controls. Some researchers have hypothesized that gut dysbiosis, perhaps triggered by factors like diet, antibiotics, or immune dysfunction, could contribute to the development or severity of autism symptoms. While this area of research is still in its infancy, there is growing interest in exploring whether microbiome-targeted interventions could help improve symptoms. Stress itself can profoundly affect the gut microbiome. Chronic stress alters gut motility, increases intestinal permeability, sometimes called leaky gut, and changes the composition of the microbiome, often reducing beneficial bacteria and allowing opportunistic species to proliferate. This stress-induced dysbiosis can, in turn, exacerbate inflammation and affect brain function, creating a vicious cycle. Breaking this cycle may require addressing both the psychological and physiological aspects of stress, including supporting a healthy gut microbiome through diet, probiotics, and stress management techniques. It's important to note that while the gut-brain axis is a rapidly growing field, much of the research is still preliminary. Most studies have been conducted in animals, particularly mice, and translating these findings to humans is not always straightforward. Humans are far more complex, both genetically and environmentally, and what works in a mouse model may not work in people. Additionally, the microbiome is just one of many factors that influence mental health. Genetics, life experiences, social environment, and brain chemistry all play critical roles, and the microbiome should be seen as part of a larger picture rather than a single cause or cure. Nevertheless, the evidence is mounting that the gut microbiome is a significant player in mental health and well-being. It's a reminder that the mind and body are not separate, but deeply interconnected. The ancient wisdom that gut feelings influence our emotions and thoughts may have a real biological basis. As we continue to explore the gut-brain axis, we may discover new ways to support mental health through the microbiome, from dietary interventions to targeted probiotics to fecal microbiota transplants. In the next section, we'll turn our attention to another critical role of the microbiome: metabolism, metabolic allies, how microbes help you digest and thrive. Every time you eat, you're not just feeding yourself, you're feeding trillions of microbes that live in your gut, and in return, they help you extract nutrients, synthesize vitamins, and process compounds that your own body cannot handle. The gut microbiome is a metabolic powerhouse, contributing a vast array of enzymatic capabilities that complement and extend your own digestive and metabolic functions. Tonight, we'll explore how gut bacteria act as metabolic allies, breaking down food, producing essential nutrients, and influencing your energy balance and overall health. Let's start with dietary fiber, the complex carbohydrates found in plant foods that human digestive enzymes cannot break down. When you eat fruits, vegetables, whole grains, legumes, and other plant-based foods, much of the fiber passes through your stomach and small intestine largely intact. It's only when this fiber reaches your colon that it encounters the bacteria capable of fermenting it. This fermentation is one of the most important functions of the gut microbiome. Different types of fiber support different bacterial communities. Resistant starch, inulin, pectin, cellulose, and hemicellulose are all fermented by various gut bacteria, including species from genera like Bacteroides, Prevotella, Ruminococcus, Bifidobacterium, and Faecalibacterium. These bacteria possess the enzymes needed to break the chemical bonds in these complex carbohydrates, releasing sugars that they can metabolize for energy. In the process, they produce short-chain fatty acids, particularly acetate, propionate, and butyrate. These short-chain fatty acids, or SCFAs, are not waste products. They are valuable metabolites that have profound effects on your health. Butyrate, for instance, is the primary energy source for the cells lining your colon, the colonocytes. About 70% of the energy needs of these cells comes directly from butyrate. This SCFA helps maintain the integrity of the intestinal barrier, reducing the risk that harmful bacteria or toxins will leak through the gut lining into the bloodstream. Butyrate also has anti-inflammatory properties, influencing immune cell function and helping to maintain a balanced immune response in the gut. Propionate and acetate, once absorbed, travel through the bloodstream to other parts of the body. Propionate is primarily metabolized in the liver, where it can influence glucose production and lipid metabolism. Some research suggests that propionate may help regulate appetite and improve insulin sensitivity. Acetate, the most abundant SCFA, can be used as an energy source by various tissues, including muscle and brain. It also plays a role in regulating cholesterol synthesis. Together, these SCFAs represent a significant source of energy, providing roughly 5 to 10% of your daily caloric intake, though this varies depending on how much fiber you consume. Beyond fiber, the gut microbiome also helps break down other complex nutrients that escape digestion in the small intestine. Proteins, for example, can be partially fermented by gut bacteria, particularly when protein intake is high or when carbohydrates are limited. This protein fermentation produces a different set of metabolites, including branched-chain fatty acids, ammonia, amines, phenols, indoles, and sulfur compounds. Some of these metabolites, like ammonia, can be harmful in high concentrations, but others, like indoles, can have beneficial effects, such as strengthening the gut barrier and modulating the immune system. The balance between carbohydrate fermentation, which produces beneficial SCFAs, and protein fermentation, which can produce potentially harmful compounds, depends on your diet. A diet rich in fiber promotes beneficial fermentation, while a diet low in fiber and high in protein can shift the balance toward less favorable metabolic processes. The gut microbiome also plays a role in fat metabolism, though this is less well understood than carbohydrate and protein metabolism. Bile acids, which are produced by the liver from cholesterol and help digest fats, are modified by gut bacteria. Primary bile acids are secreted into the intestine, where they emulsify fats and aid in their absorption. In the colon, bacteria convert primary bile acids into secondary bile acids through a process called deconjugation and dehydroxylation. These secondary bile acids can have signaling functions, interacting with receptors in the gut and liver that regulate metabolism, inflammation, and even glucose homeostasis. Some secondary bile acids promote health, while others may contribute to disease, highlighting the importance of a balanced microbiome. One of the most remarkable metabolic contributions of the gut microbiome is the synthesis of vitamins. Your body cannot produce certain essential vitamins on its own, and while you obtain most of these from your diet, your gut bacteria can also synthesize them. The microbiome is particularly important for producing vitamin K and several of the B vitamins. Vitamin K is essential for blood clotting and bone health. There are two main forms: vitamin K1, found in green leafy vegetables, and vitamin K2, or menaquinone, which is produced by bacteria. Gut bacteria, particularly species from the genera Bacteroides and Escherichia, synthesize menaquinone. Studies with germ-free animals have shown that without gut bacteria, vitamin K deficiency can develop, leading to impaired blood clotting. Humans on very low vitamin K diets, who are also treated with broad-spectrum antibiotics, which suppress the gut microbiome, can show decreased vitamin K levels and clotting problems, demonstrating that microbial production of this vitamin contributes to our overall vitamin K status. The B vitamins are a group of 8 water-soluble vitamins involved in numerous metabolic processes, including energy production, DNA synthesis, and nerve function. These include thiamine, B1, riboflavin, B2, niacin, B3, pantothenic acid, B5, pyridoxine, B6, biotin, B7, folate, B9, and cobalamin, B12. Gut bacteria can synthesize several of these vitamins, and studies have estimated that they may produce up to 30% of the recommended daily intake for the host, though this depends on the composition of the microbiome and the individual's diet. Folate, also known as vitamin B9, is particularly important. It's involved in DNA replication, repair, and methylation, as well as the synthesis of nucleotides and certain amino acids. Some gut bacteria, especially species of Bifidobacterium and Lactobacillus, can produce folate. High folate-producing strains have been identified, and studies in rats and humans have shown that administering these strains can increase fecal folate levels, suggesting that microbial folate production can contribute to the host's folate status. This has led to interest in developing probiotic strains that can help prevent folate deficiency, which is associated with anemia, birth defects, and other health problems. Cobalamin, or vitamin B12, is another vitamin that some gut bacteria can produce, though its synthesis requires a complex, energy-intensive pathway involving about 30 enzymatic steps. Vitamin B12 is crucial for nerve function and red blood cell formation, and humans typically obtain it from animal-based foods. While gut bacteria in the colon can produce vitamin B12, it's absorbed primarily in the ileum, the lower part of the small intestine, which is upstream from the colon. This means that much of the bacterially produced B12 may not be efficiently absorbed. Nevertheless, the microbiome's ability to produce B12 could still have local effects in the gut and may contribute to the vitamin status of the host under certain conditions. Other B vitamins, including riboflavin, thiamine, and biotin, are also synthesized by gut bacteria, though the extent to which this contributes to the host's nutritional status varies. The presence of bacterial genes for vitamin biosynthesis has been confirmed through metagenomic studies, and the abundance of these genes can change with age, diet, and health status. For example, folate synthesis genes are more enriched in the gut microbiomes of infants, while cobalamin and thiamine biosynthesis genes increase with age. Populations from different geographic regions also show variation in the abundance of vitamin biosynthetic pathways, reflecting differences in diet and lifestyle. The gut microbiome's role in metabolism extends beyond just producing nutrients. It also influences how efficiently you extract energy from food, a factor that can affect body weight and metabolic health. Studies comparing germ-free mice to conventional mice with normal microbiomes have shown that the conventional mice extract more calories from the same amount of food. Germ-free mice actually need to consume up to 30% more food to maintain the same body weight as their conventionally raised counterparts. This is because the microbiome breaks down dietary components that would otherwise be indigestible, making additional energy available to the host. This has implications for obesity. Research has shown that the composition of the gut microbiome differs between lean and obese individuals. In one influential study, the gut microbiomes of obese mice were found to be enriched in genes involved in harvesting energy from food. When germ-free mice were colonized with microbiota from obese mice versus lean mice, the mice receiving the obese microbiota gained more weight, even when consuming the same amount of food. This suggests that the microbiome can influence energy balance and weight gain. In humans, similar patterns have been observed, though the relationship is complex and influenced by many factors, including diet, genetics, and lifestyle. The gut microbiome also interacts with the metabolism of drugs and other xenobiotics, foreign compounds that enter the body. Bacteria can modify drugs before they are absorbed, affecting their efficacy and toxicity. For example, the drug sulfasalazine, used to treat inflammatory bowel disease, is converted by gut bacteria into its active form. Similarly, the cancer drug irinotecan is reactivated by bacterial enzymes in the gut, which can cause severe diarrhea as a side effect. Understanding how the microbiome metabolizes drugs is an emerging area of research that could lead to more personalized medicine, where treatments are tailored based on an individual's microbiome composition. Another fascinating aspect of microbial metabolism is the processing of dietary polyphenols, plant compounds found in foods like fruits, vegetables, tea, coffee, and red wine. Polyphenols are often touted for their antioxidant and anti-inflammatory properties, but many of them are not well absorbed in the small intestine. Instead, they reach the colon, where gut bacteria break them down into smaller metabolites that can be absorbed and exert beneficial effects. The ability to metabolize certain polyphenols varies between individuals, depending on their gut microbiome composition. This is one reason why the health benefits of polyphenol-rich foods can differ from person to person. As we reflect on the metabolic contributions of the microbiome, it's clear that these tiny organisms are doing far more than just living inside us. They are actively participating in our physiology, extending our metabolic capabilities, and influencing our health in profound ways. They help us extract energy and nutrients from food, synthesize essential vitamins, regulate our immune system, and even affect our brain function. Without them, we would struggle to survive. In the next section, we'll explore one of the most critical relationships between the microbiome and the host, its role in training and regulating the immune system. Training your immune system. Microbes as teachers. From the moment you are born, your immune system faces an enormous challenge: how to distinguish between the trillions of harmless or beneficial microbes that colonize your body and the small number of pathogens that pose a genuine threat. This is no simple task. The immune system must learn to tolerate the commensal microbiota while remaining vigilant and ready to respond to invaders. The gut microbiome plays a central role in this education, acting as a trainer that teaches the immune system what to attack and what to ignore. Tonight, we'll explore how the microbiome shapes the development and function of the immune system, influencing everything from the maturation of immune cells to the prevention of inflammatory diseases. The relationship between the microbiome and the immune system is bidirectional. The microbiome influences the immune system, and the immune system, in turn, shapes the microbiome, maintaining the balance between tolerance and defense. This partnership begins in early life and continues throughout your lifetime, with consequences for your susceptibility to infections, allergies, autoimmune diseases, and even cancer. Let's start with the development of the immune system in infancy. When a baby is born, their immune system is still immature. Many of the immune cells are present, but not yet fully functional, and the immune system has not yet learned to recognize and respond appropriately to the vast array of microbes it will encounter. The early colonization of the infant's gut by bacteria is critical for immune maturation. Studies with germ-free mice, which are born and raised in completely sterile conditions, without any microbes, have revealed just how important this microbial exposure is. Germ-free mice have severely underdeveloped immune systems. Their gut-associated lymphoid tissue is smaller and less organized than that of conventional mice. They have fewer immune cells, including T cells, B cells, and innate immune cells, like macrophages and dendritic cells. The T cells that are present are skewed in their function, with a reduction in certain subsets, like T helper type 1 cells, which promote cell-mediated immunity, and T helper type 17 cells, which help defend against extracellular bacteria and fungi. Germ-free mice also have reduced levels of immunoglobulin A, or IgA, the antibody that is secreted into the gut lumen to bind and neutralize bacteria and toxins. These defects can be reversed by colonizing germ-free mice with normal gut bacteria. Once bacteria are introduced, the gut-associated lymphoid tissue expands, immune cell populations normalize, and IgA production increases. This demonstrates that the presence of the microbiome is necessary for the proper development of the immune system. But not all bacteria have the same effect. Different bacterial species and strains can elicit different immune responses. Some promote the development of regulatory T cells, which help maintain tolerance and prevent excessive inflammation. Others induce the differentiation of T helper 17 cells, which are involved in defense against pathogens, but can also contribute to inflammation if not properly regulated. One bacterium that has been extensively studied for its effects on the immune system is segmented filamentous bacteria, or SFB. In mice, SFB colonizes a specific niche in the small intestine and induces the development of T helper 17 cells. These cells produce inflammatory cytokines, like interleukin 17, which help recruit other immune cells and enhance the barrier function of the gut. While T helper 17 cells are important for defense, they can also contribute to autoimmune diseases if they become overactive. Interestingly, SFB has not been found in humans, but similar bacteria may play analogous roles in shaping human immune development. Another group of bacteria that profoundly influences the immune system are those that produce short-chain fatty acids. As we discussed in the previous section, SCFAs like butyrate, propionate, and acetate are produced when gut bacteria ferment dietary fiber. These SCFAs have potent effects on immune cells. Butyrate, for example, promotes the differentiation of regulatory T cells, which suppress inflammation and maintain immune tolerance. It does this by influencing gene expression in T cells through epigenetic mechanisms, specifically by inhibiting enzymes called histone deacetylases. Butyrate also has direct anti-inflammatory effects on other immune cells, including macrophages and neutrophils, reducing their production of pro-inflammatory cytokines. The gut microbiome also influences the production of EGA, the most abundant antibody in the body. EGA is secreted into the gut lumen, where it coats bacteria, preventing them from adhering to the intestinal epithelium and penetrating deeper into the body. EGA helps maintain a peaceful coexistence between the host and the microbiota by neutralizing potentially harmful bacteria without triggering a strong inflammatory response. The production of IgA is dependent on signals from the microbiome. Certain bacteria and bacterial products stimulate the differentiation of B cells into IgA-secreting plasma cells. In germ-free mice, IgA levels are dramatically reduced, demonstrating the importance of microbial stimulation. Beyond the gut, the microbiome also influences immune function at distant sites. Microbes and their metabolites can travel through the bloodstream or send signals that affect immune cells in the skin, lungs, liver, and even the brain. For example, SCFAs produced in the gut can influence the function of immune cells in the respiratory tract, affecting susceptibility to respiratory infections and allergic airway disease. This concept, sometimes called the gut-lung axis, suggests that a healthy gut microbiome can protect against respiratory illnesses. The skin microbiome also plays a role in local immune function. Skin commensals help train the immune cells of the skin, teaching them to distinguish between harmless residents and potential pathogens. Some skin bacteria produce antimicrobial compounds that directly inhibit the growth of pathogenic bacteria. For example, Staphylococcus epidermidis, a common skin commensal, can secrete molecules that inhibit Staphylococcus aureus, a pathogen that can cause skin infections. The presence of a healthy, diverse skin microbiome can thus provide a form of colonization resistance, preventing pathogens from gaining a foothold. The microbiome's influence on the immune system is not limited to infancy. Throughout life, the microbiome continues to interact with the immune system, helping to maintain homeostasis and respond to challenges. However, there appears to be a critical window in early life during which the immune system is particularly plastic and responsive to microbial signals. Studies in mice have shown that the ability to fully restore immune function by colonizing germ-free mice with bacteria is limited to a short period in early life. if colonization is delayed until adulthood, some immune defects cannot be fully corrected. This suggests that early life microbial exposure is crucial for setting the immune system's baseline function, a concept sometimes referred to as the window of opportunity. Disruptions to the microbiome during this critical window can have lasting effects on immune function and disease susceptibility. For example, early life exposure to antibiotics has been linked to an increased risk of allergies, asthma, and obesity in numerous epidemiological studies. While antibiotics are sometimes necessary and life-saving, their overuse, particularly in infancy, may have unintended consequences by disrupting the developing microbiome and altering immune programming. The hygiene hypothesis, first proposed in the late 20th century, suggests that reduced exposure to microbes in overly sanitized environments may contribute to the rise of allergic and autoimmune diseases in developed countries. The idea is that the immune system evolved to be constantly exposed to a diverse array of microbes, and when this exposure is lacking, the immune system may become dysregulated, leading to inappropriate responses to harmless allergens, or even to the body's own tissues. While the hygiene hypothesis is still debated, and its mechanisms are not fully understood, it underscores the importance of microbial exposure for healthy immune development. The microbiome also plays a role in the immune system's ability to fight cancer. The immune system can recognize and destroy cancer cells, a process known as immune surveillance. However, tumors often develop mechanisms to evade the immune system. In recent years, immunotherapies, treatments that boost the immune system's ability to attack cancer, have revolutionized cancer treatment. Remarkably, the gut microbiome has been found to influence the effectiveness of these immunotherapies. Studies have shown that patients with certain gut bacterial compositions respond better to immune checkpoint inhibitors, a type of immunotherapy that blocks proteins that prevent the immune system from attacking cancer cells. For example, high levels of bacteria like Akkermansia, Mucinifera, and certain species of Bifidobacterium have been associated with better responses to these therapies. Researchers are now investigating whether modulating the microbiome through probiotics or fecal microbiota transplants could improve cancer treatment outcomes. On the flip side, an imbalanced or dysbiotic microbiome can contribute to immune dysfunction and disease. Chronic inflammation driven by gut dysbiosis has been implicated in a wide range of conditions, including inflammatory bowel disease, rheumatoid arthritis, type 1 diabetes, and multiple sclerosis. In inflammatory bowel disease, for example, there is often a reduction in beneficial bacteria and an overgrowth of potentially harmful species. This imbalance can trigger an excessive immune response against the gut microbiota, leading to chronic inflammation and tissue damage. Treatments aimed at restoring microbial balance, such as probiotics, prebiotics, and fecal microbiota transplants, are being explored as potential therapies for these conditions. The microbiome's role in training and regulating the immune system highlights just how interdependent we are with our microbial partners. The immune system does not develop or function normally without the microbiome, and the microbiome, in turn, is shaped and controlled by the immune system. This partnership is a product of millions of years of co-evolution, a delicate dance that maintains health and prevents disease. In the next section, we'll explore what happens when this balance is disrupted, leading to dysbiosis and illness. When balance breaks, dysbiosis and health. Throughout this journey, we've seen how the microbiome contributes to digestion, metabolism, immune function, and even mental health. But what happens when the microbial balance is disrupted? The term for this state is dysbiosis, an imbalance in the microbial community, characterized by a loss of beneficial species, an overgrowth of harmful or opportunistic species, or a reduction in overall diversity. Dysbiosis is not a disease in itself, but it is increasingly recognized as a contributing factor to a wide range of health conditions. Tonight, we'll explore how dysbiosis develops, what effects it can have, and the conditions it has been linked to. Dysbiosis can arise from many causes. One of the most common is antibiotic use. Antibiotics are designed to kill bacteria, and while they are essential for treating bacterial infections, they do not discriminate between harmful pathogens and beneficial commensals. A single course of broad-spectrum antibiotics can drastically reduce microbial diversity in the gut, sometimes for weeks or even months. Repeated or prolonged antibiotic use can have even more lasting effects, potentially preventing the full recovery of the microbiome. This loss of diversity and disruption of microbial communities can create an opportunity for opportunistic pathogens to proliferate. One well-known consequence of antibiotic-induced dysbiosis is infection with Clostridioides difficile, commonly called C. diff. This bacterium can cause severe diarrhea and colitis. It's normally present in low numbers in many people's guts, kept in check by the surrounding microbial community. But when antibiotics wipe out much of that community, C. diff can multiply unchecked, producing toxins that damage the gut lining. C. diff infections can be difficult to treat, as they often recur even after antibiotic therapy. One of the most effective treatments for recurrent C. diff infection is fecal microbiota transplantation, or FMT, where stool from a healthy donor is transferred to the patient to restore a balanced microbiome. FMT has success rates of over 90% for C. diff, demonstrating the power of restoring microbial balance. Diet is another major factor that influences the microbiome and can lead to dysbiosis. A diet low in fiber and high in processed foods, refined sugars, and unhealthy fats can reduce microbial diversity and shift the composition of the microbiome towards species that thrive on simple sugars and fats, rather than complex carbohydrates. These shifts can reduce the production of beneficial SCFAs and increase the production of pro-inflammatory metabolites. Over time, a poor diet can contribute to chronic low-grade inflammation and metabolic dysfunction, increasing the risk of obesity, type 2 diabetes, cardiovascular disease, and other conditions. Conversely, a diet rich in diverse plant foods, whole grains, legumes, nuts, and seeds supports a more diverse and resilient microbiome. The variety of fibers and polyphenols in these foods feeds different bacterial species, promoting a balanced community. Fermented foods like yogurt, kefir, sauerkraut, kimchi, and kombucha can also contribute beneficial bacteria to the gut. While the bacteria from fermented foods may not permanently colonize the gut, they can still have transient beneficial effects, interacting with the resident microbiota and immune system. Chronic stress is another factor that can disrupt the microbiome. Stress activates the hypothalamic pituitary adrenal axis, leading to the release of stress hormones like cortisol. These hormones affect gut physiology, altering motility, secretion, and permeability. Stress can also directly alter the composition of the gut microbiome, reducing beneficial bacteria and allowing opportunistic species to expand. In animal studies, exposure to stress has been shown to decrease levels of lactobacillus and increase levels of potentially harmful bacteria. This stress-induced dysbiosis can, in turn, contribute to gastrointestinal symptoms like abdominal pain, bloating, and diarrhea, creating a feedback loop where stress worsens gut health and gut dysfunction exacerbates stress. Environmental exposures, including pollutants, heavy metals, and pesticides, can also impact the microbiome. Some studies have found that exposure to environmental toxins can reduce microbial diversity and alter community composition, though more research is needed to fully understand these effects. Lifestyle factors such as physical activity, sleep patterns, and even the use of certain medications beyond antibiotics, including proton pump inhibitors and non-steroidal anti-inflammatory drugs, can influence the microbiome as well. Dysbiosis has been implicated in a wide range of diseases. One of the most well-established associations is with inflammatory bowel disease, or IBD, which includes Crohn's disease and ulcerative colitis. People with IBD often have reduced microbial diversity, with a decrease in beneficial bacteria like Faecalibacterium prausnitzii and an increase in potentially harmful species. The exact cause of IBD is still not fully understood, but it's thought to involve a combination of genetic susceptibility, immune dysfunction, and environmental factors, with the microbiome playing a key role. The dysbiotic microbiome in IBD is both a consequence of the disease, driven by inflammation and altered gut physiology, and a contributing factor, as the imbalanced microbiota can perpetuate inflammation. Irritable bowel syndrome, or IBS, is another gastrointestinal condition associated with dysbiosis. IBS is a functional disorder characterized by abdominal pain, bloating, and altered bowel habits, without obvious structural abnormalities. While the causes of IBS are complex and likely multifactorial, studies have found differences in the gut microbiome of people with IBS compared to healthy controls. Some research suggests that an overgrowth of bacteria in the small intestine, a condition called small intestinal bacterial overgrowth, or SIBO, may contribute to IBS symptoms in some patients. Treatments targeting the microbiome, including probiotics and dietary interventions like the low FODMAP diet, have shown promise in reducing IBS symptoms. Beyond the gut, dysbiosis has been linked to metabolic disorders. Obesity, for example, is associated with a less diverse gut microbiome and shifts in the relative abundance of certain bacterial phyla. Studies have found that obese individuals often have a higher ratio of firmicutes to bacteroidetes, though this pattern is not universal. The microbiome of obese individuals may be more efficient at extracting energy from food, contributing to weight gain. In animal studies, transferring the microbiome from obese mice to germ-free mice can induce weight gain, even when the mice consume the same amount of food as controls receiving lean microbiomes. While the relationship between the microbiome and obesity is complex and influenced by many factors, including diet, genetics, and lifestyle, it's clear that the microbiome plays a role in energy balance and metabolism. Type 2, diabetes, and metabolic syndrome are also associated with gut dysbiosis. People with these conditions often have reduced microbial diversity and alterations in the production of SCFAs and other metabolites. Some studies have found that certain bacterial species, like Akkermansia mucinifera, are reduced in people with type 2 diabetes, while others are increased. Restoring or modulating the microbiome through diet, probiotics, or other interventions is being explored as a potential strategy to improve metabolic health. Allergic diseases, including asthma, eczema, and food allergies, have also been linked to early life dysbiosis. As we discussed earlier, the mode of delivery, antibiotic exposure, and diet in infancy can all influence the developing microbiome, and disruptions during this critical period may increase the risk of allergic diseases. The mechanism is thought to involve improper immune training, where the absence of certain key bacteria prevents the immune system from developing appropriate tolerance to harmless antigens, leading to allergic sensitization. Autoimmune diseases, where the immune system mistakenly attacks the body's own tissues, have also been associated with dysbiosis. Conditions like type 1 diabetes, multiple sclerosis, rheumatoid arthritis, and lupus have all shown connections to altered gut microbiomes in various studies. While the exact mechanisms are still being investigated, it's thought that gut dysbiosis may contribute to immune dysregulation, increasing the permeability of the gut barrier and allowing bacterial products to enter the bloodstream, where they can trigger or exacerbate autoimmune responses. Even cardiovascular disease has been linked to the gut microbiome. Certain gut bacteria can metabolize dietary choline and carnitine, found in red meat and eggs, into a compound called trimethylamine, or TMA. TMA is then absorbed into the bloodstream and converted by the liver into trimethylamine N-oxide, or TMAO. Elevated levels of TMAO have been associated with an increased risk of heart disease, stroke, and death. Some researchers hypothesize that TMAO contributes to cardiovascular disease by promoting atherosclerosis, the buildup of plaques in arteries. However, the relationship between TMAO and heart disease is still debated, and more research is needed to understand causality. Cancer is another area where the microbiome is being intensively studied. As mentioned earlier, the microbiome can influence the effectiveness of cancer immunotherapies. But the microbiome may also play a role in cancer development itself. For example, Fusobacterium nucleatum, a bacterium associated with colorectal cancer, has been found to promote tumor growth and suppress anti-tumor immunity. Other bacteria have been linked to gastric cancer, liver cancer, and other malignancies. Understanding these connections could lead to new strategies for cancer prevention and treatment. Mental health disorders, as we explored in the gut-brain axis section, are also associated with dysbiosis. Depression, anxiety, autism, and other psychiatric conditions have all shown alterations in the gut microbiome in various studies. While the microbiome is not the sole cause of these conditions, it appears to be one contributing factor, and restoring microbial balance may offer a complementary approach to traditional treatments. It's important to recognize that dysbiosis is not always the primary cause of these diseases. In many cases, it's a contributing factor or a consequence of the disease process itself. Inflammation, altered gut physiology, medications, and other aspects of illness can all affect the microbiome. This makes it challenging to determine cause and effect. Is the dysbiosis driving the disease, or is the disease causing dysbiosis? In reality, it's likely a bidirectional relationship, with dysbiosis and disease influencing each other in a vicious cycle. Despite these complexities, the concept of dysbiosis has important implications for treatment. If microbial imbalance contributes to disease, then restoring balance could help alleviate symptoms or even prevent disease. This has led to growing interest in microbiome-targeted therapies, which we'll explore in the next section as we discuss how to nurture and support a healthy microbiome. Nurturing your microbiome: diet, lifestyle, and future possibilities. Now that we've explored the many roles of the microbiome and the consequences of dysbiosis, you might be wondering, how can I support a healthy microbiome? What can I do to nurture these microscopic allies and reap the benefits they offer? Tonight, we'll discuss practical strategies for promoting a diverse, balanced microbiome through diet and lifestyle. And we'll also look ahead to the future of microbiome science, including emerging therapies and interventions. Let's start with diet, which is arguably the most powerful tool you have for shaping your microbiome. The food you eat doesn't just nourish you, it also feeds your gut bacteria, and different foods support different microbial communities. A diet rich in diverse plant foods is one of the best ways to promote microbial diversity and abundance of beneficial bacteria. Fiber is the cornerstone of a microbiome-friendly diet. Dietary fibers are complex carbohydrates that resist digestion in the small intestine and reach the colon, where they are fermented by gut bacteria. Different types of fiber feed different bacteria, so consuming a variety of fiber sources is key. Whole grains like oats, barley, and brown rice, legumes like beans and lentils, fruits, vegetables, nuts, and seeds all provide different fibers that support different bacterial communities. Prebiotics are a specific type of dietary fiber that selectively promote the growth of beneficial bacteria. Common prebiotics include inulin, found in foods like chicory root, garlic, onions, and asparagus, and fructooligosaccharides, found in bananas, artichokes, and wheat. Resistant starch, found in foods like cooked and cooled potatoes, green bananas, and legumes, is another type of prebiotic. These compounds are not digested by your own enzymes, but are readily fermented by gut bacteria, promoting the production of SCFAs and supporting a healthy gut environment. Polyphenols, the plant compounds found in colorful fruits and vegetables, tea, coffee, cocoa, and red wine, are also beneficial for the microbiome. While many polyphenols are not well absorbed in the small intestine, they reach the colon, where gut bacteria metabolize them into smaller compounds that can have anti-inflammatory and antioxidant effects. Foods rich in polyphenols include berries, apples, grapes, green tea, dark chocolate, and extra virgin olive oil. Including a wide variety of these foods in your diet supports a diverse and resilient microbiome. Fermented foods are another excellent addition to a microbiome-friendly diet. Fermentation is a process in which bacteria or yeast break down sugars and produce acids, gases, or alcohol, transforming the food and often making it more digestible and nutritious. Foods like yogurt, kefir, sauerkraut, kimchi, miso, tempeh, and kombucha contain live bacteria and yeasts that, while they may not permanently colonize your gut, can still interact beneficially with your resident microbiota and immune system. Regular consumption of fermented foods has been associated with increased microbial diversity and improved gut health. On the flip side, certain dietary patterns can harm the microbiome. A diet high in processed foods, refined sugars, unhealthy fats, and low in fiber can reduce microbial diversity and promote the growth of bacteria associated with inflammation and metabolic dysfunction. Excessive consumption of artificial sweeteners has also been shown in some studies to negatively affect the gut microbiome, though more research is needed. Red meat, particularly when consumed in large quantities, can lead to the production of TMAO, a compound linked to cardiovascular risk, as discussed earlier. Reducing or eliminating unnecessary antibiotic use is another important step in protecting your microbiome. While antibiotics are sometimes necessary and life-saving, they should be used judiciously and only when truly needed. If you do need to take antibiotics, consider supplementing with probiotics during and after the course to help restore microbial balance, though you should discuss this with your healthcare provider. Probiotics are live microorganisms that, when consumed in adequate amounts, can confer health benefits. They are available as dietary supplements and are also found in fermented foods. Different probiotic strains have different effects, so choosing the right probiotic depends on your specific health goals. For example, Lactobacillus rhamnosus GG is one of the most well-studied strains and has been shown to help prevent and treat diarrhea, including antibiotic-associated diarrhea. Bifidobacterium species are often beneficial for gut health and may help alleviate symptoms of IBS. Saccharomyces boulardii, a beneficial yeast, can help prevent C. diff infections. However, it's important to note that not all probiotics work for everyone, and the quality and viability of probiotics can vary between products. Prebiotics and probiotics can also be combined in what are called synbiotics, products that contain both a prebiotic substrate and a probiotic organism, theoretically enhancing the survival and colonization of the probiotic. Lifestyle factors beyond diet also influence the microbiome. Regular physical activity has been associated with increased microbial diversity and higher levels of beneficial bacteria, including those that produce SCFAs. Exercise may promote microbial health through multiple mechanisms, including alterations in gut transit time, changes in immune function, and shifts in metabolic activity. Even moderate amounts of exercise, such as walking or cycling for 30 minutes most days of the week, can have positive effects. Sleep is another important factor. Disrupted or inadequate sleep has been linked to alterations in the gut microbiome, and conversely, the microbiome may influence sleep quality through the production of neurotransmitters and other signaling molecules. Maintaining a regular sleep schedule and getting 7 to 9 hours of quality sleep per night supports not just your microbiome, but your overall health. Stress management is also crucial. As we discussed, chronic stress can negatively impact the microbiome, and the microbiome can influence the stress response through the gut-brain axis. Practices that reduce stress, such as mindfulness meditation, yoga, deep breathing exercises, and spending time in nature, can benefit both your mental health and your gut health. Exposure to diverse environments and microbes, particularly in childhood, can also promote a diverse microbiome. Growing up in homes with pets, spending time outdoors, and even playing in the dirt can introduce a variety of microbes that may benefit the developing microbiome and immune system. The hygiene hypothesis suggests that some exposure to microbes is necessary for healthy immune development, though this must be balanced with the need to prevent infectious diseases. Looking to the future, microbiome science is rapidly advancing, and new therapies are on the horizon. Fecal microbiota transplantation, or FMT, which we mentioned in the context of C. diff infections, is being explored for other conditions, including inflammatory bowel disease, obesity, and even autism. FMT involves transferring stool from a healthy donor to a patient, with the goal of restoring a balanced microbiome. While it has shown remarkable success for C. diff, its effectiveness and safety for other conditions are still being investigated in clinical trials. Next-generation probiotics, sometimes called live biotherapeutics, are being developed. These are carefully selected strains of bacteria, or even consortia of multiple strains, that are designed to target specific health conditions. Unlike traditional probiotics, which are often based on strains found in fermented foods, these next-generation probiotics are identified through rigorous scientific research and tailored to specific therapeutic goals. Some of these therapies are already in clinical trials for conditions like IBD, metabolic disorders, and even cancer. Another emerging area is the use of microbial metabolites as therapeutics. Instead of introducing live bacteria, researchers are exploring the use of the beneficial compounds that bacteria produce, such as SCFAs, to directly modulate health. For example, butyrate supplements are being studied for their potential to improve gut health and reduce inflammation. Personalized nutrition based on the microbiome is also an exciting frontier. Researchers are working to develop algorithms that can predict how an individual's microbiome will respond to different foods, allowing for personalized dietary recommendations that optimize health. Some companies already offer microbiome testing services that claim to provide personalized diet and lifestyle advice, though the scientific validity and utility of these tests are still debated. As our understanding of the microbiome deepens, it's becoming clear that this hidden universe inside us holds tremendous potential for improving health and preventing disease. But it's also important to approach microbiome science with caution and critical thinking. The field is still young, and while we've learned a great deal, there is much we don't yet understand. Not every claim about the microbiome is backed by solid science, and the microbiome is just one of many factors that influence health. That said, the principles of supporting a healthy microbiome, eating a diverse, plant-rich diet, getting regular exercise, managing stress, avoiding unnecessary antibiotics, and nurturing your microbial partners are rooted in sound science and align with broader recommendations for healthy living. By caring for your microbiome, you're not just supporting these microscopic organisms, you're supporting your own health, resilience, and well-being. As we bring our journey through the microbiome to a close, take a moment to reflect on the incredible complexity and beauty of this hidden ecosystem. Trillions of microbes, each one a world unto itself, working together in an intricate dance that sustains your life. They are ancient partners, evolved alongside us, woven into the very fabric of what it means to be human. In caring for them, we care for ourselves, and in understanding them, we gain a deeper appreciation for the interconnectedness of all life. Conclusion. Tonight, we've traveled through the universe inside you, exploring the vast and intricate world of the microbiome. We've seen how trillions of microorganisms colonize every surface of your body, from your skin to your gut, forming communities that are as diverse and complex as any ecosystem on Earth. We've learned how these microbes contribute to digestion, synthesize vitamins, train your immune system, communicate with your brain, and influence nearly every aspect of your health. We've discovered how the microbiome is seeded at birth, shaped by diet and lifestyle, and how disruptions can lead to dysbiosis and disease. And we've explored how you can nurture your microbiome through simple, practical steps, supporting these microscopic allies in their work. The story of the microbiome is a story of partnership, a reminder that we are not solitary beings, but superorganisms, living in symbiosis with a vast community of microbes. They are not invaders or passengers. They are integral parts of who we are. As science continues to unravel the mysteries of the microbiome, we are gaining new insights into health, disease, and the fundamental nature of life itself. As you drift off to sleep tonight, think about the incredible hidden universe within you. Imagine the trillions of cells, both human and microbial, working in harmony to keep you alive and well. Let this knowledge fill you with a sense of wonder and appreciation for the complexity and beauty of your own biology. And remember, you have the power to support this inner ecosystem, to nurture it and help it thrive through the choices you make every day. Thank you for joining me on Bedtime Biology tonight. If you enjoyed this exploration, please like this video, subscribe to the channel for more journeys into the quiet wonders of the natural world, and leave a comment below sharing your thoughts or where you're checking in from. Sweet dreams, and may your microbiome flourish as you rest.